Friends, Namaste and welcome to Dr. Varsha Gaitonde's channel. In this video, I am going to discuss some of the questions on plant breeding statistics and biometrical genetics. Let us discuss one by one. The first question is father of mutation breeding. The four options given are Seth, Gustafson, Muller and Morgan. Here the answer is Gustafson. Let us see the contribution of three more scientists. Seth Wright, he worked with the male lamb. He found the mutation for the first time. Then Muller's work was to discover the mutagenic action of X-rays in Drosophila. For that work only he got Nobel Prize in the year 1946. Then Morgan's work was in linkage and sex linkage. He did work with Drosophila genetics, sex linkage and linkage, that is development of theory in genes. For his enormous work, he got now first Nobel Prize in genetics in the year 1933. The second question is for screening quantitative traits, most preferred population in mutation breeding is, the answer is M3, because if we are screening for the quantitative traits, here the trait identification itself is very difficult. So for that, bit uniformity is very essential. So we may go for M3 generation. If instead we are taking qualitative traits, means we should screen in the M2 generation. Third question is North Carolina 1 design is also called as. North Carolina 1 design is a biparental mating design. It is known as nested design. Okay. Factorial design is North Carolina design 2, means North Carolina design 2 is also called as factorial design. Then let us see what is compact design and mixed design. The compact design is nothing but split plot design or lattice design kind. We shall discuss all the experimental designs in a separate video. Here just you understand that compact design is nothing but just kind of split plot design or lattice kind design. Then mixed model. Mixed model is conceptualizing the complex correlation structures in the breeding. Next question, father of cytogenetics. The options given are Boveri, Jensen, Sutton and Morgan. Answer here is Sutton. How about the other three scientists' contribution? Boveri. Boveri is a person who is a co-founder of modern cytology. Jensen, he is the discoverer of the compound microscope, organized compound microscope. Then Morgan, as I have already discussed, he contributed for linkage and sex linkage. Next question is Go-Go method of crossing technique is preferred in. Go-Go method is practiced in wheat. Here I am giving you one assignment. What are the different crossing techniques preferred in rice, tomato, mustard? You should enlist and keep it in comment section. Okay. Next question is GP4 includes. The options given are related species, wild ancestors, transgenes and advanced lines. Answer here is transgenes. Why? Because gene pool 4, here mostly unrelated families or all the unrelated plant materials are placed. Plant material, animal material, whatever the living things are there, they are placed in GP4. How about GP1, GP2, GP3? Gene pool 1 where all the related species are placed. Okay. Gene pool 2 means wild species, wild ancestors, which may be partially crossable. And gene pool 3 means somewhat difficult to cross. Where these advanced lines are placed, these are placed in gene pool 1 or 2. Means there is a overlapping. So let us see what are the advanced lines. Advanced lines are the plants whose germplasm has been manipulated from the parental material from which the new variety of hybrids are developed. That means by little altering, hybrids are developed. Next question. Significance of difference among several means is tested with the help of. The options are chi-square test, t-test, f-test and z-test. Answer is f-test. Okay. So let us see where this chi-square, t-test and z-test are used. Chi-square test is used to compare the two variants in a contingency table to see if there are relation or differences. Means in Mendelian population, to differentiate the observed frequency and expected frequency, generally we use chi-square test. Second is t-test. Where this t-test is used, it is used where the hypothesis is used to determine if there is a significant difference between the means of two groups from the parental material. There the f-tests are used, t-test is used, and when the sample size is less than 20, where there t-tests are used. And how about z-test? Here when the sample size is more than 50, to compare the things in a population, z-test is used. Next question is most serious error is. The options are type 1, type 2, both, none. The answer is type 1 error. Type 1 error is the error where we are rejecting the true things. Okay. It is also called as false positive, and type 2 error is called false negative. That means in type 1 error we are rejecting the null hypothesis when it is true, and in type 2 it is vice versa. Next question, most effective number of composite lines in synthetic population in field crops are. Remember, here it is asked in case of field crops. Okay. The answer here is 5 to 10. If forage crops are preferred means 10 to 20 lines are also taken. Okay. In field crops only, if the plant population is large, we may go for 6 to 10 lines also. But in this case, when exclusively field crops are asked, the answer is 5 to 6. Don't get confused. If forage crops are asked means 10 to 20. Okay. Next, most preferred marker for detection of rare alleles. Four options are given here: SSR, SNP, RFLP and RAPD. Answer is SNP, that is single nucleotide polymorphism. This is a kind of dominant marker, and RAPD is also a dominant marker here, whereas SSR and RFLP are codominant marker. SSR is simple sequence repeats, RAPD is random amplified polymorphic DNA, and RFLP is that is a kind of codominant marker. Restriction fragment length polymorphism is the full form of RFLP. Next is active resistant mechanism operates in response to the. Options are pathogen infection, pathogen dimorphism, host resistance and host avoidance. The answer here is pathogen infection. The other options, pathogen dimorphism. The meaning of dimorphism is reproduction in different forms. This is majorly observed in case of fungi, where host tolerance means in case of presence of the pathogen invasion or presence of pathogens only, they tolerate the infection and perform better. Host avoidance means avoiding in any form, maybe physical, cultural or any kind of host mechanisms. Next question is, Will is a rust resistant variety of. The options are barley, wheat, oats and groundnut. The answer is barley. Here I am again giving one more assignment to you. You just enlist the rust resistant varieties in wheat, oat and groundnut and keep it in the comment section. Next question is, most utilized database site is. Here 4 databases are given: Swissprot, EMBL, MDB and NCBI. MDB is Maze database. Then the other one is EMBL, European Database Library, Swissprot, and the last one is NCBI, that is National Center for Biotechnological Information. The most preferred one is NCBI. Okay, and this is having most of the information, all databases. Next question is, multiline breeding was popularized by. The 4 options are Swaminathan, Venkataraman, Borlaug and K.C. Mehta. The answer to multiline breeding is Borlaug. Okay, he developed several multilines and he gave different varieties also. The other options are Swaminathan, Venkataraman, K.C. Mehta. We shall enlist their contributions. Swaminathan, as you know, the father of green revolution. He brought several wheat and rice varieties into India, and he doubled, tripled the food production. To many times he increased. He is a reason to increase the food production. Then Venkatraman was a sugarcane breeder. Then K.C. Mehta, he was a discoverer of Puccinia path in India. He has partially contributed for gene deployment method also. Next question is, heterosis is a potent source of practical gains was proposed by. The four options are Shull, Hull, Sprague and Tatum. Answer is Shull. What is the contribution of Hull? Hull is, he has given the concept SCA in the year 1945, and Sprague and Tatum, they combinedly proposed the combining ability theory in the year 1942. Next question, average performance of a strain in a series of crosses is. The four options are GCA, SCA, heterosis and heterobeltosis. When average performance is asked, it is GCA. When specifically it is asked, then specific combining ability. Heterosis, you know, the progeny performance better in one or the other character from the parent. Heterobeltosis means betterment of the progeny over the best parent. Next is most widely used male sterility system is four options: CMS, TGMS, transgenic sterility, CGMS. CMS is cytoplasmic male sterility, TGMS is temperature genetic male sterility, then transgenic sterility is also one of the kind of genetic male sterility, then CGMS, which is a three-line system. Okay, most widely used one is CGMS. Here three lines are used. It is time consuming as well as space consuming, but most preferred as well as most efficient method of genetic male sterility. Okay, the next question is the triticale variety released from India. Popular variety is TL 419. All are the triticale varieties only, but the variety released in India is TL 419. The natural inducer gene of haploidy in maize. Four options are KR1, KR2, IG and ID. There are no ID genes, I think. IG genes are found in maize, and KR1 and KR2 are the natural haploid inducer genes in wheat. Next question and last question. Number of chromosomes in cotton endosperm. As you know, endosperm is always having 3N ploidy number. Then if in case of cotton, as you know, cotton Gossypium hirsutum is a tetraploid. The chromosome number of it is 52. If we'll take the basic chromosome number or X, it is 13. Okay, 13 means here 3N. N is 13 into 3 is 39. So the answer is 39. Friends, hope the questions I discussed here are helpful for you. If you have any doubt, please keep it in the comment section, and don't forget to make your homework. Thanks once